Every cell in your body contains a molecule so extraordinary that it holds the complete instruction manual for building and running an entire human being. This molecule, DNA, is only 2 nanometers wide, yet if stretched out from a single cell would measure approximately 2 meters long. Inside every one of your 37 trillion cells sits this same complete instruction set, copied perfectly billions of times. But how does DNA actually work? DNA stands for deoxyribonucleic acid. Its structure is a double helix, 2 long strands twisted around each other like a spiral staircase. Each strand is made of repeating units called nucleotides. Every nucleotide contains 3 components: a sugar molecule called deoxyribose, a phosphate group, and one of 4 nitrogen bases: adenine, thymine, guanine, and cytosine, abbreviated as A, T, G, and C. The 2 strands connect through these bases following strict pairing rules. Adenine always pairs with thymine, and guanine always pairs with cytosine. These base pairs are held together by hydrogen bonds, weak individual bonds, but collectively strong enough to hold the entire structure together. A single human DNA molecule contains approximately 3.2 billion of these base pairs, arranged in a precise sequence that carries all genetic information. This sequence of base pairs forms genes, specific segments of DNA that carry instructions for building proteins. The human genome contains approximately 20,000 genes, though genes make up only about 2% of total DNA. The remaining 98% was once called junk DNA, but is now understood to play crucial regulatory roles, controlling when and how genes are expressed. The process of reading genetic instructions and building proteins happens in 2 stages. The first stage is transcription. When a gene needs to be expressed, an enzyme called RNA polymerase binds to a specific region of DNA called the promoter. The double helix unwinds, and the polymerase reads the DNA template strand, building a complementary messenger RNA molecule, mRNA, using the same base pairing rules, except uracil replaces thymine in RNA. This mRNA strand carries the genetic message out of the cell nucleus. The second stage is translation. The mRNA travels to a ribosome, the cell's protein building machinery. The ribosome reads the mRNA sequence in groups of 3 bases called codons. Each codon corresponds to a specific amino acid. Transfer RNA molecules, tRNA, carry amino acids to the ribosome, matching their anticodon sequence to the mRNA codon. The ribosome links amino acids together in the precise order specified by the genetic code, building a protein chain that folds into its functional 3-dimensional shape. DNA replication ensures genetic information passes accurately to new cells. The enzyme helicase unwinds and separates the double helix. DNA polymerase then reads each strand and builds a new complementary strand alongside it, always adding nucleotides in the 5' to 3' direction. The result is 2 identical double helix molecules from one original. This process copies 3.2 billion base pairs with extraordinary accuracy, making only approximately 1 error per billion base pairs copied, with additional proofreading mechanisms correcting most errors immediately. Mutations occur when errors in DNA replication or damage from radiation, chemicals, or other factors alter the base sequence. Most mutations are harmless or repaired immediately by dedicated DNA repair enzymes. Some mutations alter protein function, occasionally beneficially driving evolution, sometimes harmfully causing diseases, including cancer, when mutations affect genes controlling cell division. DNA is tightly packaged inside the nucleus by wrapping around protein spools called histones, coiling further into structures called nucleosomes, then condensing into the familiar X-shaped chromosomes, visible during cell division. Humans have 46 chromosomes, 23 pairs, containing our complete genome. Modern biotechnology can now read entire genome sequences in hours, edit specific DNA sequences using tools like CRISPR-Cas9 with extraordinary precision, and even synthesize artificial DNA sequences. Capabilities transforming medicine, agriculture, and our fundamental understanding of life itself. Thanks for watching my video. I hope you enjoyed it.